Hi guys, just a quick video to talk about Cairn's technique for measuring the length of DNA molecule with autoradiography by Vivi and Yi. Okay, so let's start. Now first is about the autoradiography. It is a bioanalytical method. Then this technique is using X-ray film to visualize molecules or fragments of molecules that have been radioactively labeled. Then this technique is used to determine the delocalization of radioactive substances in the particular cells. And finally, it is normally used in biological experiments. After the introduction, now I would like to mention about the principle of autoradiography. In autoradiography, the radioisotopes will emit the radiation. So you will be questioning what is radioisotopes. Then later on in Cairn's technique, it will be explained clearly to you all. Then the radiation then causes the photographic film to be ionized. After that, the photographic film is a mixture of silver halide, and gelatin. And in this technique, a radioactive substance is put in direct contact with a thick layer of photographic emulsion, and the thickness is about 5 to 50 millimeters. And by having gelatin substances and silver halide crystal, this emulsion differs from the standard photographic film in terms of having high ratio of silver halide to gelatin and small size. Okay, let me write it. Small size of grain. Finally is the methodology of autoradiography. First, take a sample of cell of a living organism, then it is covered with a photography emulsion containing alkyl and gelatin. Place the sample in the dark room at a period of time, usually 2 months, and wait for the radiation to emit. This sample is then washed away to get insoluble silver atom only, Ag. Finally, the autoradiography can be now observed under microscope. I think that we already have an understanding of what is the autoradiography. Therefore, I will move on to Cairn's technique. So, Hugh John Foster Cairn is a British molecular biologist, and he is also the one who demonstrated that the bacterium E. coli contains a single circular molecule of DNA. And this is the image of him. Then now I would like to mention the steps, or you could say the procedure in Cairn's technique. First, cells grown for 2 generations in a culture medium containing tritiated thymidine. Okay, so now I will explain to you what is tritiated thymidine. So thymidine alone is thymine linked to a deoxyribose. And tritiated thymidine is tritium in here. And what is tritium? Actually, tritium is a radioactive isotope of hydrogen. Isotope H3. And this tritiated thymidine is radioactively marked the DNA in Cairn's technique, and specifically in this experiment, Cairn decided to use E. coli. Step 2 in Cairn's technique is when cells placed onto a dialysis membrane, cell walls digested by lysozyme. So if anyone does not know what is lysozyme, so lysozymes are glycosidases that assist in hydrolysis of glycosidic bonds between acetylmumaric acid and acetylglucosamine, and these enzymes therefore too destructive to many bacteria since they can damage bacterial cell wall that is made up of peptidoglycan. And then the surface is immersed in radioactively sensitive emulsion, which demonstrated right here, containing silver bromide AgBr. After step 2 is step 3 in the Cairn's technique, and in this step 3 is when the radiation from the tritiated thymidine converts the Ag plus ions in silver bromide into insoluble metal grains, which indicated in the graph as the black dots. In step 4 is when the thin film of photographic emulsion applied to surface left in darkness for 2 months. And in that 2 months, that is when tritiated thymidine decaying leads to tritium, the isotope of hydrogen atoms in DNA decay. Here it emits beta particles, which can be also understood as high energy electrons, and these high energy electrons produce a dot on a photographic plate. And after 2 months, the film is developed, and then it can be examined under electron microscope, which will give us the original data in Cairn's technique. And the dark green where tritium decayed, it is the indication DNA position. And through Cairn's technique, it proves that the prokaryotes, which is the E. coli used in this experiment, has a DNA which is single circular molecule, whereas the eukaryote has a linear DNA. And finally is Cairn's the original data that observed from Cairn's technique. In this observation, the strand of E. coli's single DNA is about 700 to 900 micrometers long, and the result that while DNA undergoes replication and evolves into a replication bubble, like the image you have shown, parent strand and daughter strand. Exposure. That is the end of our presentation. We hope you have a nice day, and bye.